Okay, so this is the last day before exam number 3. Exam number 3 is on Wednesday, so don't forget about all your SmartBook assignments. Because we only did 4 chapters this time, there's only 4 SmartBook assignments. And that last one is actually fairly short. So I know it was uploaded kind of late, but this chapter is kind of short. We're not covering a whole lot in it. It says, I think on average it says it should take you about 20 minutes. So it's a lot shorter than the others. But yeah, make sure you finish your 4 SmartBook assignments. I think that's all I got. Don't forget that you do need to be in here on Wednesday to take an exam. I know most of you that are here every day know that, but there are a lot of people listening to the recording that aren't here every day. But you do have to be here on Wednesday to take your exam. All right, any questions before we get started about the exam or what's going to be on it? Everybody pretty clear? Okay, so like I said, this should be a relatively short chapter. Because we're not going into a whole lot of details on a lot of this. Diagnosing infections is a very, very cool topic, and I wish we had more time to go into all of it, but a lot of it is intensely complicated, and we just don't have enough time to go through all of it. So we're going to go through kind of the basics, just so you get an idea of what the inside of a micro lab looks like in a hospital or a doctor's office, what they're generally trying to accomplish, and then we're going to move on. So chapter 15, diagnosing infections. So the whole purpose of diagnosing an infection is what is causing this person's condition, right? A patient, remember that all of this is always tied back to a person. This is always tied back to a person who is experiencing something, a patient who has a disease or a sickness, and we are trying to help them. So what is causing this condition? And there are a couple of different ways, 3 different ways, that we typically diagnose infections. And all 3 of these are still used really commonly in different ways. So the first way that we kind of diagnose infection is through phenotypic testing. And phenotypic means physical, I don't know what is going on with that pen, sorry. It is looking at the physical properties of an organism. Now, these could be things that we can see, like the shape of an organism when we look at it under a microscope, or what color it grows on a plate, or some other physical characteristics. This could also be things that are not actually visible. We're going to talk about that more in a second, but biochemical testing is typically testing for an enzyme, the presence or absence of an enzyme. I have that on a slide in a second. But those things are not necessarily visible, but we can do tests to determine whether or not they have that enzyme. So again, these are physical properties of an organism. Either it's physiological properties, like things that you can see with your eyes. Sometimes they're not things that you can see with your eyes. Sometimes it's whether or not it has an enzyme or not, which is not something that you can see physically, but it is still a physical property of that organism. It has the enzyme or it doesn't have the enzyme. The second way we test for organisms or we try to diagnose diseases are through immunologic techniques. And immunologic techniques is basically using the idea of antibodies and antigens. Okay, immunologic basically means related to the immune system. So the immune system, kind of the basis, especially for that adaptive immune system, is that antibodies, one specific antibody, will bind to one specific antigen, right? Antibodies and antigens are very, very specific. One binds with one. And so you can use that idea that antibodies and antigens are very specific for each other to test for the presence of antibodies or antigens. And you can do this both ways, and we'll look at it in just a minute in more detail. But you can you can test for the presence of antigens by using antibodies, but you can also test for the presence of antibodies by using antigens. So you can do it both ways. And that's what it says right there, analysis of microbe using antibodies or patient's antibodies using prepackaged antigens. And we'll look at that in more specific terms in just a couple of slides. But immunologic testing in general has to do with antibodies and antigens, like the immune system, immunological. And then the third way is genotypic, and genotypic is just looking at the genetic material. So it's using different testing techniques to look for the presence or absence of a specific organism's genetic material. And like I said, all three of these are still commonly used in a lot of different ways. We're going to talk about some specific examples in a bit. And what type you want to use depends on the organism, depends on the infection. Some things grow really, really well on media, and so phenotypic testing is the easiest. Some things don't grow well on media, and so immunologic testing is easier. It's just going to depend on the scenario, the patient, and the organism. But before you can do any kind of testing, the first step to any of that testing, phenotypic, immunologic, or genotypic, is collecting a specimen. So specimen collection is the first step of any diagnosis. And as most of you want to be nurses at some point in the future, this is the part of this process that is for you. The rest of it is mostly done by medical laboratory scientists in the hospital lab or the doctor's office lab or wherever you send your lab work to. And it's important that you know what's happening on the other side of those swabs and things like that. But as nurses or doctors, this is the step that you will most likely partake in. And specimen collection is just collecting of a specimen. Now, the type of sample collected is going to depend on the suspected illness, right? If you suspect somebody of having a urinary tract infection, collecting a nose swab isn't going to do much for you. Okay, if you suspect somebody of having a urinary tract infection, you probably want to collect a urine sample. If you suspect somebody of having a GI illness, right, they have something going on inside their gut, you're probably going to want to collect a fecal sample. If you suspect somebody having the flu, you're going to collect a nasal swab. So, right, whatever the infection is, whatever the suspected infection is going to determine what type of sample you will collect. And this is just a list of some of the most common specimens collected in hospitals: urine, feces, spinal tap sometimes, sputum. Sputum, if you don't know what that is, it's the mucus from deep in your lungs. So like when you're coughing and you cough stuff up, like when you're sick, that's sputum. Vaginal samples, you can get skin biopsies, all different kinds of things, okay? And like I said, it's what you collect is going to depend on the infection. However, something that all specimen collections have in common is that one of the most important pieces of specimen collection is avoiding contamination. Specifically, avoiding contamination of that patient's normal flora. So remember, when we are collecting a specimen, we are trying to diagnose an infection. And to do that, the best way to do that is to not collect that patient's normal flora, right? We don't care about their normal flora. We care about the disease that's being caused. So that's why, again, if you've ever had to give a urine sample, especially if you're a genetic female, if you're somebody that has a vagina, and you've had to give a urine specimen, they have little special wipes that you're supposed to wipe yourself down with, right? To clear away. They give you like these little, maybe you guys are too young to have been to an OBGYN before, but when you go to a new OBGYN for the first time and get a pap smear, they're gonna make you pee in a cup to give a urine specimen, and they give you these little wipey things to wipe yourself off with. And that is called a clean catch specimen because it's avoiding the natural vaginal flora, We're checking for what's in the bladder, not what's in the vagina. All that stuff is normal. We don't care about that. That's also why if you've ever had a COVID test, right, those COVID test swabs, they go way up there, right? They don't get like right in your nostril. A good COVID specimen feels like it's touching your brainstem, right? It goes way up in there because we don't care about the normal flora around your nostrils. We care about the flu or the COVID that's growing way up in your sinus cavity. So that's one of the things that all specimen collection has in common. And it's a really, really important part of being a caregiver is being able to collect specimens that are going to be useful to the lab. Okay, again, another example is if you've ever had blood collected, what do they do before they collect your blood? They wipe your skin down with alcohol, right? That's not just to prevent you from getting an infection, it's also to prevent the blood in the tube from being contaminated with your skin flora. Because if it's contaminated with your skin flora, we might get an incorrect reading. Okay. All right, so phenotypic methods. Again, phenotypic methods are the physical properties of an organism. And again, just because it's a physical property doesn't necessarily mean it's something that you can see. One of my physical properties is that I have 2 lungs, right? Can you see my lungs by looking at me? No, but I still have them. You could do tests to see them, to test to determine if I have lungs. I do. But it's the same thing with bacteria, right? You can't see everything that's on the inside of a bacteria. So physical properties don't necessarily mean something you can view with your eyes, but it's a physical property. So there's 3 kind of ways that we can do this. The first one is through direct examination of a specimen. And these are the visible characteristics. And this is usually done by viewing a specimen is usually done by viewing a specimen through a microscope, with or without the organism being stained. So one really quick way of determining if somebody has a yeast infection, vaginal yeast infection, ear yeast infection, mouth yeast infection, is to take a sample of that and put it under a microscope. Yeast are really, really big, and they're really, really easy to see. So it doesn't take any other diagnosing. You just look at it under a microscope and see the physical characteristics of budding yeast and diagnose them with a yeast infection. Okay. Another really, really common way, probably the most common way that we directly observe or directly examine specimens in a micro lab is by gram staining them. So you take the direct specimen, whatever it is, whether it's a swab from a wound or a fecal sample, or I got an entire toe in the lab one time. Whatever the sample is, you take a small piece of it, put it on a slide, and look at it under a microscope. A lot of times you gram stain it to see what color it turns. It also allows you to see the shape of the organism. So this right here is a gram stain of a sputum sample. Okay, so somebody hacked this up from their lungs, and I took a small piece of that sputum sample and put it on a microscope slide and then gram stained it. If you're in this class, you don't have to know too much about gram staining. All you have to know is that gram positive is purple and gram negative is pink. But if you're in the lab, you have to know a lot more about gram staining, right? But what I can see on this sputum sample is that, first of all these cells right here, all these kind of big pink blotchy things, those are all white blood cells, which is not normal in a lung. You usually get white blood cells when you have inflammation, when you have a sign of infection, because your white blood cells are being pulled out of the bloodstream and to the tissue where an infection is. And then I also see lots of these little purple things. Those are gram-positive cocci. Okay, so I can see what shape they are, I can see what color they're staining, I can see their arrangement. If you remember arrangements from lab, I can see that these are what we call diplococci, which means it's two circles right next to each other. Okay, so you can tell a lot from that sputum specimen. Because I have been trained, you guys don't have to know this, but because I've been trained, I know that this person probably has Streptococcus pneumoniae, just based on what I see under a gram stain. So these are all physical characteristics. It's the size of the organism, the color, the arrangement, the shape of the organism itself. Okay? These are all physical, viewable, visible characteristics. Next, we have culture and morphology. So culture and morphology is kind of just what it sounds like. It's taking that specimen, putting it on media, and seeing how it reacts to specific media after it has been incubated. Okay, there are a lot of different medias out there, and again, for this class, you don't have to know any of them. If you've taken lab or you're in lab, you most certainly have to know them. You're kind of in the middle of doing this right now, if I'm not mistaken. But certain types of media will allow certain characteristics of the bacteria to show. Okay, so certain types of media allow some organisms to grow and not others. Some organisms turn specific colors on certain media. Okay, some of them do something called hemolysis, which is what you're seeing here, which is breaking down the red blood cells in media. Right, there's a lot more information we can gather based on the visible characteristics of growing on certain culture media. And again, we're not going to talk about the specifics here. This is basically what you've spent the entire quarter learning in lab, if you're taking lab, is how certain things grow on certain types of media. And a big part of identifying microorganisms through phenotypic methods is learning what all of that different media means and how certain organisms react on certain media. Does anybody who's taking lab know what kind of hemolysis this is? I know you have to know it. Beta hemolysis, I saw it. That's beta hemolysis. Have you guys learned about McConkey augers yet? Is this a lactose fermenter or a non-lactose fermenter? That's a lactose fermenter because it's pink on McConkey. Again, if you're not in lab, you don't have to know that. But this is how we tell certain things about an organism. I know that this organism is capable of breaking down red blood cells in a blood agar. I know that this organism is a gram-negative rod and can ferment lactose, just based on the fact that it is pink on this plate. Okay, so there's a lot of things that you can tell based on certain types of media. Okay. And then the last type of phenotypic method is biochemical testing. And this is testing for all different kinds of biochemical properties. Most often, it is going to relate to sugar usage or sugar utilization and the presence or absence of certain enzymes. Again, you don't have to know for this class the specific types of biochemical tests. Most of them, again, are testing for whether an organism can use a certain sugar. So, like, for example, this organism here is a lactose fermenter. It can use lactose for food. Some organisms can't. Some organisms can only use glucose and maltose. Some organisms can only use maltose. Some can use mannitol. Some can only use sucrose, right? Each organism is going to have different properties. And that's the point of all this, okay? And then another common type of biochemical testing is testing for the presence or absence of certain enzymes. Again, something going on? The presence or absence, you guys are like all looking around. Something funny happening that I'm missing out on? All right. So if an organism has lactase, or if an organism has catalase, or if an organism has oxidase, or if an organism has urease, okay, all different kinds of enzyme tests. And again, this is not something you have to know for this class. You have to learn about it in the lab. And the idea behind all of this is that each organism is going to have a unique biochemical signature. And learning what all of those are is a huge part of medical microbiology, if you ever take that. So biochemical testing is really, really important to phenotypic identification. And the way that we typically identify certain organisms is by using something called a dichotomous key. And a dichotomous key is just a special kind of flow chart. Now keep in mind, you don't have to memorize this dichotomous key, but what you do have to understand is how to use one. And you'll notice that this is a flow chart that helps you determine a bacteria's identity based on a combination of phenotypic traits, culture traits, morphology traits, and biochemical traits. Okay? So what I mean by you need to know how to use one is, let's say I give you this dichotomous key, and I'll give you a hint. I will absolutely be giving you a dichotomous key that you then have to use, okay? If I give you this dichotomous key, again, you don't have to memorize it. I'm going to be giving it to you. But if I give you this dichotomous key, and I say you have an organism that is gram positive, catalase positive, and strictly aerobic, what is the genus of that organism? Think about it for a sec. Micrococcus, okay? Because we said that this organism was gram positive. We know that it's a cocci. It's gram positive. And then we look at the catalase next. That's catalase positive, and it's strictly aerobic, okay? And the organism there is Micrococcus. All right, what if it was gram negative, oxidase negative? Vianella. That's a weird word, but it's Vianella. It's the V one down at the bottom, okay? Because again, it's gram negative, oxidase negative, Vianella. All right, so that's all you have to need to know how to do for my class. I will tell you the results. I think for lab, typically the kind of test that you have to know is you have to know how to interpret these results. So for my class, we're not talking about gram stains or catalase. You don't know what a positive catalase is or a negative catalase is for my class, but in lab you would. So in lab, she'll show you a picture of a positive catalase reaction, and you have to know that that's catalase positive. But for my class, if I give you something like this with a dichotomous key, you have to be able to tell me what the organism is. Okay? And dichotomous keys and phenotypic methods of identification are still very, very common for most easily grown bacteria. So most bacterial infections in hospitals are tested for this way. Okay, it's just that the people testing them have all these flowcharts memorized. Okay, but most normal bacterial pathogens, people who have wound cultures or sputum cultures or stool samples or any other urine samples, all those samples are typically put on media and gram stained and tested for using these different phenotypic methods, either gram staining, looking at the direct examination, the culture and morphology, and then the biochemical testing. Okay. So again, this is still very common and most commonly used for easily grown bacteria. Most of the bacteria that grow inside your human body are very easy to culture. They take 24 hours to grow, and then you ID them using biochemical methods. Now, immunological testing, again, uses the principles of antigens and antibodies to diagnose infection. And immunological testing, not always, but it's most common for things that aren't easily grown. So things like viruses, right? Like here we have, this is a COVID test. This is an immunological test for COVID. You guys have probably all seen one, probably all have them at your house. Another really common immunological test is for hormones, like pregnancy tests, right? We diagnose pregnancy using pregnancy tests. You pee on the test. 1 line means you're not pregnant, 2 means you are. That's an immunological test. Or certain types of toxins. So these are all things that are not going to grow very well on media or at all, right? Hormones are not organisms. They're not going to survive on media. Neither are toxins. This is also how you can do, there are like drug test kits that you can buy at Walgreens. You can test your urine for drugs. And it's something very similar to this. It's an immunological test. A drug is a type of toxin or some kind of foreign substance. Again, those types of things don't grow or don't grow well. Viruses are very difficult to grow in labs. And so we don't use immunological or we don't use culture methods to diagnose viral infections, typically. So immunological testing is going to be most common for things that don't grow. Viruses, hormones, drugs, toxins, things like that. And one of the bases of immunological testing is the idea of agglutination. And I'll move it over in a second. But agglutination is basically clumping or coagulation. And agglutination happens when a specific antigen finds its specific antibody. Okay, so when you have the correct antibody antigen pair, that is going to lead to agglutination. And remember that antibodies and antigens are very, very specific. You're not going to get agglutination unless the one antibody that you're looking for binds with a specific antigen. Okay, they're individual. One antibody is always going to match up with one antigen. All right, so there are 2 general ways that this can happen. Again, like it said on the previous couple of slides, you can either use antibodies to diagnose antigens, or you can use antigens to diagnose antibodies. So those are 2 different questions. In the first question over here, the question is, does this patient make antibodies? Okay, so in this case, we are testing for, our unknown is whether or not there are antibodies. So to test for whether there are antibodies, what we do is we add antigens. Okay, so in this case, this is a prepared, known microbial antigen. This is what we call a reagent. So this is something that you have in a bottle that somebody prepared in a lab that you are then going to add to the patient's unknown serum, which is the clear part of their blood, which is where your antibodies are. So if we're testing if a patient has produced antibodies to a particular antigen, what we can do is take that serum, take that person's unknown serum, and add a known antigen to it and see if you have clumping. In a case where that person doesn't have the antibody, right, over here, they don't have that specific antibody that binds that specific antigen. So in that case, there's no antibody-antigen pair, and there will be no agglutination. So it'll just look clear. On the right, however, that person, that patient, did produce antibodies. And so because the antibody is present in their serum, it will bind to the antigen we added, and it will cause clumping, which you can see in that little drop. Okay, you can see the little chunks that have formed. They just look like little beads or little clumps. Okay? And then the second question is, what is the identity of that microbe? So in this case, what is known is, or what is unknown, is the antigen. Right? So in the first one, what was unknown was the antibody. In this case, what is unknown is the antigen. And so when you're testing for a specific antigen, you add known antibodies. And when you add the antibodies and there are no antigens present that match up, you'll have no binding. But if you add that antibody and that microbe has that specific antigen, it will bind and clump up and cause agglutination. So again, the idea here is the presence or absence of specific antigens and antibodies. All right. The way that this COVID test works is that those antibodies, so what we're testing for in a COVID test is which one? Are we testing for 1 or 2? No, in a COVID test, we're testing for the antigen. We're trying to see if that COVID virus is present in your nose. That's the purpose of a COVID test. We're not testing your antibodies, we're not testing your blood. We're testing to see if there is COVID virus growing in your nose. Okay, so we are testing, we're doing this one, question 2. What is the identity of this organism? Okay, and on these COVID tests, what they have is this testing line has embedded, that's not a good color. Okay. It has embedded antibodies in that paper. Okay, so that in paper has antibodies sitting in the paper in a straight line. And as the snot from your face travels across that piece of paper, if there are the correct antigens there, those antigens will bind to the antibody on the paper and cause clumping, which makes a visible color change. When those antigens bind to that antibody, you have a physical clumping color change. It's just on this, they have it lined up in a straight line, so it looks like a line instead of like this. Okay, again, pregnancy tests are the same thing. Pregnancy tests are testing for the beta-HCG hormone. Okay, and so it's testing for an antigen. And it has antibodies in the paper, and when the urine runs over it, if the antigen is present, it binds to the antibodies and causes a physical color change. All right. Another way of applying the same idea, and this is just taking what we were doing over here, specifically question 1, does this patient make antibodies? You can do something called an antibody titer. And what an antibody titer is it just determines the amount of antibodies a person has. So it's taking that same immunological idea from before, does this person create antibodies, and doing it over and over and over to get an idea of how many antibodies that person has. And this can be really useful for a couple of reasons. One, to determine Okay, so one of the ways that this is commonly used is to determine if a person has enough antibodies to be what we consider immune. So if you've ever heard of somebody doing this, and probably when I started working at a hospital, I had to do this. So if you ever want to be a nurse, as far as I know, If you work in any kind of healthcare field, you'll have to do what's called getting your titers done. And what your titers are is they determine how many antibodies you have to illnesses that you were vaccinated for. So when I started working at a hospital, I had to have measles, mumps, and rubella titers done from the MMR vaccine, because you're vaccinated for MMR when you're like a year old, right? And some people retain their immunity. They produce high amounts of antibodies forever. Some people don't. And so they need to be re-immunized or re-vaccinated. Okay. Again, I had to be tested for MMR, so measles, mumps, and rubella, and then I had to be tested for hepatitis B. And you have this whole series of titers that you have to get done to determine if you have enough antibodies present in your body to be considered immune. Okay. And another way that this can be used is to monitor a known disease. So remember back to when we looked at the immune response, when a person is going through the initial stages of their immune response, right, their antibody levels to that particular antigen are going to remain low, and then they're going to go up, and then eventually they're going to go back down. So if we want to figure out where in the curve that person is, we can monitor their titers over time. So we can see, oh, this week they had a titer of 140, 1 over 40. And the next week they had a titer of 1 over 160, which we'll talk about in a second, but that means that it's getting higher. Okay, so they had an increase in the amount of antibodies, so they are on the upswing. They're still in the active part of their infection. But if their antibody titers went down, then they're on the downswing. And that's most common for diseases that have long-term progressions, things like syphilis. Syphilis lasts for months. So syphilis is not something that you get and you're over in a couple of weeks. Syphilis lasts for months. And so it can be really hard to tell at what stage somebody is in when they have syphilis, because they don't always show physical symptoms. So this can be useful for determining that. Okay, and the way that we do this is we basically add decreasing amounts of serum So you just add decreasing amounts of serum, which is where the antibodies are, to the same amount of antigen. And you basically look at how little serum you can add and still get a reaction. So what we mean by that, so what we're looking at here is the concentration. So the concentration of antibody here, or the concentration of serum, is 1 to 20. That means we took 1 part serum and 20 parts liquid. Okay? So that is the highest concentration, 1 out of 20. In this one, it's 1 to 40, which means we've cut the concentration in half. We've added half the amount of serum. In this one, it's 180, which again, we've cut the amount of serum in half. So we're adding decreasing amounts of serum to each one of these tubes. And this person has a reaction all the way down to 1 over 160. Right? So you still see agglutination in this tube, but at 1 over 320, they no longer have a reaction. So this person's titer is 160. That's how we report that. It means that they have a reaction up until you dilute the serum 1 to 160. But if you dilute it any further, that is going to not have a reaction. Okay? So again, this is the highest concentration over here. So this is the largest amount of serum, and this is the least amount of serum. Okay. And if an antibody or if a reaction goes out further, so if you're able to dilute it even further, wait, never mind. This is the least serum right here. Sorry, I didn't realize we had a control. This control means that you didn't add serum. Okay. But if the antibodies, if you still have reactions, even when you keep diluting out the serum, that means that you have a higher amount of antibodies in your original serum. Okay, so say this person has a titer of 160. So a person who has a titer of you can't see that. So somebody who has a titer of 160 versus somebody who has a titer of 320, who has more antibodies? Which one? You're pointing to it. 320. This person has more antibodies. And somebody who has a titer, this thing is starting to drive me crazy, a titer of 32 is going to have less antibodies. Okay? So if a person who we suspect has syphilis comes in and has a titer 1 week of 32, and then they come in 2 weeks later and their titer is 320, are they in the active part of their disease where they're going up, or are they getting better and it's going down? Is it going up or down? The amount of antibodies. We went from 32 to 320. Is that up or down? Up. They are getting more antibodies. When they came in 1 week, they had a titer of 32. And then they came in 2 weeks later, and they have a titer of 320. That means the amount of antibody that they're producing is going up, which means that they are in the active part of their infection. All right? Now, say somebody has this person, this titer of 160. Say that they have a titer of 160 for measles. So they have their titers checked, and this is their measles titer. They have a titer of 160. Say to be considered immune for measles, you have to have a titer of greater than 64. Okay, that's what the standards say. The standards say that to be considered immune to measles, these are all made up numbers. Okay, don't quote me on this. But if I say that somebody has to have a titer of 64 to be considered immune to measles, is this person immune to measles? Yes, because their titer is higher than 64. Okay? As the amount of serum goes down, so that's what we're doing here, is we're diluting out the serum. So the amount of serum is going down. The further out you see reactions indicates that you had more antibodies. All right. So those are ways that we commonly use immunologic methods to test for diseases. And the last one we're just going to cover very briefly. Again, this is another one of those chapters. In your book, there are tons and tons of tests. They talk about ELISA and Western blots and microarrays and all different kinds of really cool technology. We just don't have the time, and this isn't the place to cover it. All right? So again, if you're reading your textbook and you're like, what is all this stuff? You don't have to know it if we don't talk about it. So genotypic methods are using PCR or other related types to test for organism genetic material. And remember, PCR is a test to replicate, or it's a procedure to replicate genetic material. It can be DNA or RNA in a tube or in a lab or in a microarray, but it basically means outside of the organism. So you take a tiny amount of an organism's DNA and you replicate it over and over and over and over again. And genotypic methods, we kind of touched on this when we talked about PCR at the end of the last section, but genotypic methods are becoming much more popular. The reason that they're becoming so popular is because they are incredibly sensitive and incredibly accurate, and they are very, very fast. So most of these methods take a couple of hours tops. Most of them take like 45 minutes. 45 minutes to an hour, and they are more accurate than most of our other tests. They're incredibly sensitive, incredibly accurate. They're very quick. The biggest downside is that they are also very, very expensive. Okay? And one example of a genotypic method that has become much more popular in the last couple of years, and again, just so that you have an idea of how quickly this is developing, is we didn't have anything like this when I started working. I started working in hospitals like 8 years ago. It hasn't been that long. We didn't have anything like this until probably 4 years ago. And we were the first people, I worked at St. Francis, we were the first people in the area to get what's called a BioFire, which is a type of genetic testing. We were the first ones to get it, and it's only been like 3 or 4 years, and now everybody in the area has one. Okay, so they're growing immensely. As you guys move through your careers, the amount of genetic testing and genotypic testing that you're going to see is going to increase exponentially. I know it. So this is going to be a big part of your training and learning how these work and what these mean. But this is an example of what's called a BioFire, which is just a brand of genotypic test. And this one specifically is called a respiratory panel. And what this is you can take a sample from someone's nose, so a swab you stuck up somebody's nose, and put it in a test cartridge for a BioFire, and in 45 minutes, it's gonna tell you whether or not this person has, how many different illnesses? 1, 2, 3, 4, 21 different illnesses. So in a matter of 45 minutes, you can tell if this person has or doesn't have 21 different illnesses. Some of these are viruses, most of them are viruses that don't get cultured, that they're very difficult to test for. And then these others down here, I know you don't know what those mean particularly, but Bordetella pertussis is whooping cough, Mycoplasma pneumoniae, we're gonna talk more about that later. Those are very difficult organisms to grow. They don't grow on typical media. And so in 45 minutes, you know that this person has COVID and influenza A. And so you can treat them appropriately very, very quickly. And so genotypic methods are really cool. They're going to do a lot in the upcoming years, and they're just going to get better and cheaper. But as of right now, the difficulty is cost, because a lot of insurance companies still don't cover these. When I left St. Francis, an individual BioFire costs like $380. And most insurance companies don't cover them. So you have to be kind of picky about when you run them, because they cost too much money, and asking somebody to pay $400 to find out that they have the flu, you know, that's a big ask. If somebody's kid is sick and you can't figure out why they're wrong, why they're sick, and maybe you think that they have mycoplasma, maybe it's worth it. But as these become cheaper, it's going to be interesting and amazing to see what things can happen. All right, so that's all I have for you. That's all the material that we're going to cover for exam number 3.